Hi Bio 30. I'm going to do a quick lecture. Well, it might not be as fast as I'd like it to go because it takes a little while to explain this. But before the long weekend, I'm going to do DNA replication. And then I might hammer off a couple more lectures tomorrow just to get it done. But I'm going to talk about how DNA actually forms into two new DNA strands. My slides kind of go from slides 124 to 131, addressing DNA replication. I'm not going to spend a lot of time on my slides today though. I'm going to put a bunch of stuff up on my chalkboard wall, and then I'll kind of summarize it after. But a term you need to know for DNA replication is that DNA is replicated in a semi-conservative fashion. So remember that this happens during the S phase of interphase for meiosis or mitosis. But before we figured out how DNA replicated, we had three competing theories, whether an original strand created a brand new helical structure from it, or if we created part of new strand, old strands with new strands dispersed within it. That's called dispersive replication. Right, so the original strand is fully conserved, and we can make completely new ones. Dispersive, where new strands are dispersed within old strands, or whether or not DNA, half of the old strands were conserved in the replication process, and we built one new template. Excuse me. Off of the original template. It turns out through experimentation that DNA, what it does is it replicates in a semi-conservative fashion. Okay, and this will make sense as this lecture goes. So replication has three main steps: initiation, elongation, and termination. I'm going to remind you that accessing those videos that I told you to look up in the previous lecture, look at the Craig Savage DNA replication video, supplemental to what I'm going to teach here, because it's really going to make these things very, very clear. Okay, so my notes talk about a bunch of things. I'm going to talk about a bunch of enzymes, and we'll talk about the stages of replication and the way it proceeds, how it ends. So initiation. So there's information here about initiation, and then building DNA strands. Okay. So all this, right? So all this terminology is in my notes. So make sure you go over that. Okay, we'll hit a terminator sequence in the replication machinery, and then the replication machinery will stop. So this will make more sense when I talk about transcription and translation, but you have start sequences or start DNA triplets, and you have stop triplets, which initiate replication and end replication. When we talk about making RNA, those are called codons. But I'm not going to talk about that much today. I'm just going to talk about the process. So, the important things that you need to know in the replication machinery are these terms in the slides, okay? So once you kind of review this, go back to the slide and look at it. I'm going to add a few things onto this as well. But I'm not going to talk about transcription and translation today. Okay, so DNA replication. So this is called this lesson is like a Milk My Student's Mind lesson. So just be aware of that. So what do we start with first? We start with a double helix, right? So if we have a helical structure, right? So remember, DNA exists as a double helix. The first thing that needs to happen in order to replicate DNA is, right, so this is a 3D structure. So we need to turn the 3D staircase, or the 3D spiral staircase, into a 2D stepladder. All right, so DNA becomes a 2D stepladder. Now there is, okay, so there's an enzyme called DNA gyrase that does that. Okay, so DNA gyrase, it eases the tension on the double helix, and it turns 3D helix into a 2D stepladder. Okay, so DNA gyrase, first thing that we need to know. Helix into a 2D structure. Now, what we have to do next is we have to break up the hydrogen bonds in the middle of the DNA strand, because we're going to add nitrogenous base pairs to each of the strands that we're separating to build a new strand on top of it, a complementary strand. All right, so DNA. We have an enzyme that comes down through the DNA and unzips the double helix. So unzips the helix, and a DNA helicase does that, or a complex of helicase enzymes. Okay, so we have the helicase that breaks up the hydrogen bonds between each complementary pair. And then you keep the helix separated by these things called SSBPs. And these are called single-stranded binding proteins. All right, so we ease the tension on the helix. We break the stepladder up into the component parts by separating the hydrogen bonds. Gyrase is our first enzyme. Helicase is the next one. So I'm going to take this helix and I'm going to invert it 90 degrees. So we're going to look at the strand like this. Okay. So this is what actually happens: is the DNA helicases, they move down the DNA strand and open up the helix at certain parts. So think of this open part kind of like a bubble moving down a track, and this is actually called the replication bubble. So this doesn't all happen at once. It happens in stages as the DNA gets coded. So the replication bubble, and on either end is a replication fork. Okay, so replication fork, replication fork. Okay, so you have both sides of the DNA strands that are now separated. Okay, so remember that the DNA is structured in anti-parallel. And one end has the hydroxide end oriented in one direction, right? So the hydroxide end here, and the phosphate head is oriented in that direction. So you'll have a 3' to 5' end and a 5' to 3' end, okay? So this one will have the phosphate heads right side up, and this side will have the hydroxide ends upside down. Okay, so what we're going to do is the replication machinery is going to, again, we separate the strands, and then we're going to add new strands inside the replication bubble that are complementary to the strands we just separated. So we're not going to make identical strands, we're going to build a single complementary strand off of the original strand. So when we make daughter cells, they contain half of the original DNA. Okay? So, what we do first is, there has to be a proofreading mechanism. Okay? So, if there's a strand that's built, if this is 3' to 5', the strand that's going to be, the new strand that's going to be built off of this strand is going to be 5' to 3'. Okay? So, what happens first is there's a proofreading mechanism. So there's an enzyme, a series of enzymes called primases. And these primases, what they do is they actually, okay, so these primases are, they are enzymes that add RNA primers. And what they do is they proofread the DNA, so check it out for mistakes, and they build a base for replication. Because we can't build the strand off nothing, so this kind of builds a base. It's sort of like, right, if I've got to paint the house or something, I've got to add, right, I've got to scrape and prep, and then I've got to prime that area first. I can't just start painting. Okay, so the primers are added in a 5' to 3' direction. Here, so these are RNA primers, so they're base pairs of RNA. And so this strand here is going to be the strand built on the other strand. Is built in the 3' to 5' direction, and it actually can't be built continuously. So if the replication machinery is overall moving this way, okay, so if we're moving that way, so if that is the direction of replication, okay, you can still only add base pairs in a 5' to 3' direction. So it's built, we'll talk about this more when we get to the next stage, but this one is built in stages. But RNA primers are still added in a 5' to 3' direction. Okay, so the RNA primers are added here, and they proofread, okay? But again, the overall direction of replication is here. All right, now once the primers have come in and everything's okayed, everything looks good, and the replication can proceed, what will happen is a series of enzymes called DNA helicases. Okay, the helicases will come in and they will remove the RNA primers. Sorry, not DNA helicases, DNA polymerases. So there's a complex of DNA polymerase enzymes. There's poly A1, poly A2, poly A3. You don't need to worry about that though. Okay, so the DNA polymerases, what they will do is they will remove the RNA primers and they will build the new complementary strand in a 5' to 3' direction. Okay, so the DNA polymerases they'll remove the RNA primers, and then the strand that is in the 5' to 3' orientation will be built continuously. Okay, So this will be on the 5' to 3' strand, complementary strand, like I said here. This will be a continuous build because the enzymes work in a 5' to 3' direction. And this is called the leading strand because it gets coated first as a result. Okay. So that's the leading strand. Now, again, remember that the direction of replication overall is this way. But the other strand itself has a 3' to 5' orientation, the other complementary strand that's being built. So it can't actually be built continuously. What happens is the DNA helicases, they come in and they remove the RNA primers and they build in chunks. So it's still built this way, but what happens is they remove the primer, build backwards, then the machinery pops to the next series, builds backwards, pops to the next series, builds backwards, pops to the next set of base pairs. Okay? So this will eventually build the new strand. Alright? So the 3' to 5' strand is built discontinuously. This is called a discontinuous build. And that's called the lagging strand because it's built a little slower. All right? And these strands that, these DNA, these nitrogen base pairs that are added by the DNA polymerases, these are called Okazaki fragments. Okay, these are called Okazaki fragments, named after the guy who discovered them. Okay, so those are Okazaki fragments. All right, so we're almost done here. Okay. Now, what needs to happen is these Okazaki fragments need to be annealed or bonded together. Okay. They can't just stay like that. So, at these spots here, you have another enzyme. And this is called DNA ligase. Okay? And what DNA ligase does, okay, DNA ligase anneals or bonds Okazaki fragments together. And any base pairs that aren't bonded on the leading strand as well, it'll bond together. So the DNA ligase bonds. So you guys saw what DNA ligase does when we talked about genetic engineering in your bioethics project, right? We're going to talk about that more when we talk about genetic engineering at the end of this unit, but that is what the ligase does. Okay, it bonds things together. So let me give you a little flow chart for this again, just to summarize. And then that will be it. And my chalkboard is sufficiently colorful for the weekend. Like I said, I'm going to try to finish up the lecture series. I'd like to do it tomorrow, but it just depends on how motivated I am. I am also getting a little tired and might need a few days worth of break. But anyway, so let's think about the stages. So I'll give you a little, you have the diagram that you can look at, but here's a flow chart. So the first thing that needs to happen, DNA gyrase, right, turns 3D helix into 2D stepladder. Okay. 2 DNA helicase. Unzips the double helix, meaning that the hydrogen bonds are separated. Right, so the nitrogen base pairs are separated, so H bonds are broken and the complementary strands are separated. Complementary strands separated. Okay, so DNA helicase will do that. Okay, DNA gyrase, DNA helicase. The next thing that we need to do that we're going to consider are primases. Okay, so primases proofread the DNA and they set up so the primases, they proofread the DNA and they set up a base for replication. Okay. All right, so that's what primases do. After that, DNA polymerases build strands in a 5' to 3' direction. Okay, so what that means is the leading strand is built continuously in 5' to 3'. So the leading strand is built continuously in 5' to 3'. And I'm running out of space here. 6, the lagging strand is built as Okazaki fragments in a discontinuous fashion because it has a 3' to 5' orientation. Okay. So the lagging strand is built as Okazaki fragments in a discontinuous fashion. And then 7, DNA ligase anneals Okazaki fragments together. And new strands. Okay, so let me just let you take a look at this again. So it's a little bit messy, but you can see it. Okay, so step 1, step 2, step 3, step 4, step 5, step 6, and then step 7. So this is simplified in comparison to what you're going to learn in university, but it is the same basic structure. There'll just be pieces added onto it. Okay, so what happens after, if I go back to my original diagram, I showed you that, right? I showed you that the DNA, the original strands, the parent strands are yellow, and the new strands that are being built are green. So what we end up with at the end of DNA replication is this. Okay, we will end up with a DNA strand that is one of the original strands. Okay. And you'll end up with the new complementary strand. Remember, they're not identical, they're complementary. Okay. And those will turn into new DNA, right? And it'll curve back into a double helix. And again, because half of the original strand is conserved, it's called semi-conservative replication. Okay, and I will remind you that this happens, semi-conservative replication, and this happens during the S phase of interphase. Okay, and that is DNA replication. All right, so that should shore up everything, pretty much everything that you need to know for your DNA replication lab. The only other thing I'm going to include, I'm just going to erase this step here. I'm going to give you the central dogma of DNA replication and protein synthesis. Okay, so what we need to know about DNA replication and protein synthesis is it always happens the same way. That's why it's called dogma, because it's always a consistent rule. All right, so this central, right, science doesn't have dogmatic thought, but in some cases there are things that always happen consistently. So we'll call it that. Of DNA replication and protein synthesis. Okay. Central dogma of DNA replication and protein synthesis. So, what happens is we always have DNA replication, and what happens is, right, is you start with an original strand and make 2 DNA strands. Right, so we turn from this and then get 2 of these. Okay, so DNA replication is DNA that always makes 2 DNA, and it happens in the nucleus. Always. It never doesn't happen in the nucleus. Okay, so step 1 is always in the nucleus. Okay. 2 is transcription. And transcription is DNA to RNA, and RNA is single-stranded, right? And that also happens in the nucleus. Never happens anywhere else, okay? So 2 also happens in the nucleus. All right? From transcription, we leave the nucleus and go to the cytoplasm. So this is the nucleus of a cell, if you didn't already figure that out, and then the outside of the cell obviously is the cytoplasm. All right, the outside of the nucleus is the cytoplasm. That's where all the organelles hang out, right? So 3 is translation. And translation is RNA, and we use the ribosome. So we use the ribosome to make protein. Okay, so that's translation, and that is in the cytoplasm. So this always happens this way. It never doesn't happen this way. Okay. And that is DNA replication. That's also your introduction to transcription and translation. I will expand on transcription and translation likely tomorrow. Anyway, have a good weekend, and we will talk to you after the long weekend. Bye now.